Okay, so I hope you enjoyed the session with Alex. So now it's my great pleasure to introduce the next keynote speaker, who unfortunately cannot be with us in person today. It's Núria López-Bigas, who probably does not need too much introducing to this audience. So Núria is a research professor at the IRB in Barcelona. She started her scientific career on this campus at the EBI as a postdoctoral fellow in 2006, and after which she then moved to Barcelona to start a research group working on cancer genomics. Núria is very well recognized for her seminal works on mutational processes, where she has investigated the genomic topography of mutations along the genome. She's also developed some of the best performing tools for cancer driver gene discovery. And as of 2 years ago, she's now co-leading a CRUK Grand Challenge that focuses on how normal cells maintain their phenotypes despite the widespread presence of driver gene mutations. So Núria will join us on Zoom, she already has. The presentation is pre-recorded, but she will be available for questions thereafter. Thank you, and I'm looking forward to your talk, Núria.
Hello, everyone. Good afternoon, and you will excuse me that I don't put my camera today. I'm not in my best shape, and that's the reason why I couldn't travel, which I regret very much, because I was looking forward to attend the conference and see many of you. But thanks to the organizers for adapting and allowing me to give this online talk. So I will talk about clonal selection in normal tissues and its relation to cancer risk. Somehow is explaining a bit the trajectory that led us to work on this project, Prominent. And the objective of this Cancer Grand Challenge project is understanding cancer promotion to inform prevention. We want to measure clonal selection in normal tissues and ask these two questions. How does the clonal selection relate to cancer risk? And how does clonal selection relate to exposures to carcinogens and risk factors? So I will explain the trajectory that led me and our lab to work on this. And part of this trajectory goes through the analysis of tumors. So I will start by saying kind of the obvious, right? There is a continuous evolution in our tissues. From the first division, there are mutations that occur generating variation between the cells in our body. And there is also a process of selection. Some mutations provide a selective advantage, leading to clonal expansions. So this is a continuous Darwinian evolution. And we call driver mutations these mutations that confer this selective advantage, leading to these clonal expansions. As a result, our body is a mosaic of clones of different sizes. Some mutations that occur very early in development may occupy a high proportion of our body. That's what we call the mosaic. But other mutations that occur later on or expanded later on also create clones, smaller clones that may be confined in only one tissue and one lineage. We have not been able until very recently to study these clones in normal tissue, but for quite a long time now, about 15 years, we have been able to study one extreme case of these clonal expansions that are tumors. And there are many projects that have sequenced tumor DNA, whole genomes and exomes, like TCGA, the Cancer Genome Atlas, International Cancer Genome Consortium, Hardwick, and many other initiatives. And in all these cases that I will use the data today, they sequence a tumor sample and a blood sample, or a second sample as to be used as germline control. And this is very often blood. This way, you can identify germline variants and distinguish them from somatic mutations from the tumor that are the ones that are exclusive of the tumor DNA. This This has been, for the last 15 years or so, being applied to many tumors. At the moment, in Intogen, that is a project that we run in the lab, we have 33,000 tumors, for which we have at least exome data, and always 2 samples. And when you look at the somatic mutations in a tumor, you can feel a bit like when you go to Rome, that you see all these ruins, all these temples, all these stones, and that looks all like a big mess, that it's not easy to grasp and understand. But if you, or archaeologists with their expertise, analyze it with the right technologies, they can actually reconstruct the history of the city very accurately. Similar feeling we have when we look at the somatic mutations in a tumor. There are thousands of mutations in one tumor. If we sequence the whole genome, and there is a total mess, or it looks like a total mess. But analyzing it with the right technology, we can do something like molecular archaeology and reconstruct at least parts of the history of these cells. In particular, we can reconstruct the history of exposures of these cells, exposures that generate mutations, mutagens, and we can reconstruct part of these clonal expansions and positive selection. Okay, so in particular, these are the 2 questions that we ask to the somatic mutations in tumors for the moment. That is, how have these mutations occurred, which are the mutational processes in those tissues and tumors, and which of these mutations are drivers. The first part is about the variation, how do mutations occur, and you know that our cells are exposed to a number of DNA damaging agents, and we have a number of DNA repair pathways that can repair lesions created by exposures to external mutagens or internal mutagens, endogenous mutagenic factors, and also mismatches. These DNA repair are very effective, but are not perfect, and this is the way that a number of mutations end up in our genomes in every cycle. Different mutational processes have a very different pattern, leave a very different pattern of mutation. It's like a footprint. And this is what are the mutational signatures that you know about. What we ask in our lab is, what is the effect of chemotherapies in our DNA? This is an important question for several reasons. One of the reasons is because chemotherapies, in addition to having side effects during the treatment, they also have late side effects, right? Like increased number of, increased risk of second malignancies, or cardiovascular problems, and other problems that we would like to do some steps to understand a bit better. This is particularly important for childhood cancer, because childs are heavily treated with chemotherapy. Many of them are cured, but they also have a long life to live ahead of them. And understanding these late side effects is important. So the first opportunity that we had to advance on that was analyzing metastases of tumors that we had whole genome. This was Hardwick Medical Foundation data. And we knew each of these patients to which treatment they were exposed to, they received. And we could identify a number of mutational signatures in the metastases due to the treatment. I'll show you just 2 examples here, capecitabine and cisplatin. But a big question that we had is, okay, this is metastasis, but because chemotherapy are a systemic treatment, they may also affect, of course, the rest of the tissues. It's not that easy to study the effect of chemotherapies in other tissues, first because of accessing to the tissue, right? We access tissues, metastases or tumors, because they are obtained for a clinical biopsy, but usually we don't get access to a kidney or a liver unless it's not usually needed. And then there is another reason, in addition to accessing to the tissue, and is that even if we would have access to the tissue, there has not been a clonal expansion. Like in the case of a tumor, that it's kind of easy to identify mutations. every cell will be different, and every mutation will be more or less private to one cell or a low number of cells. So it poses also a difficulty in identifying the mutations. We had the opportunity recently, this is a paper that we published a couple of months ago, to study childs that had, during their life, two independent tumors. This is one of the cases. In the paper, there are other interesting things. I'm just going to focus to these results. This is a child that developed neuroblastoma when she was 1 year and 5 months old. She was heavily treated with chemotherapy, and she was cured. She had normal life, and when she was 9 years and 11 months, she was diagnosed with a malignant rhabdoid tumor, apparently totally independent tumor, and she died. The parents gave access to the autopsy very generously for research, so we could access to these tissues, DNA from these tissues, that we solved kind of a first difficulty to answer the question. But as I said, we also had to apply special techniques, and in this case, we did duplex sequencing in this DNA to see what was the effect of the chemotherapy in the DNA of these normal tissues. General result, you can read the paper because it's now out, is that we can identify the mutational footprint of the chemotherapy in all the tissues, and the number of mutations, this is number of mutations per day of treatment, is not low, on the order of 10 mutations per day, and usually they are treated for a good number of days. And this is as compared to the aging mutations per day. So I think many of the people in the audience know what is duplex sequencing, but just to explain it very briefly, because I will talk about it later as well. This is a technique that allows you, by barcoding, identify the two strands of the same molecule, sequence them both, and this way you can identify true mutations, because if you see a T in one strand, you have to see an A in the other strand, from PCR errors, sequencing errors that are plenty. So this allows to identify very rare mutations. Even if we have seen it only in one molecule, we can trust them. And in this case was important because all these mutations are very private. The other thing that is important when we see the variation or how the mutations occur is that there is variability in the mutation rate along the genome. Some regions of the genome accumulate larger number of mutations and some others much lower number of mutations. This is important because this means that different sites have different probability to mutate. Different regions of the genome, but also different trinucleotides based on the mutational signature. And this is very important to model neutral mutagenesis. I'm sorry. One thing that we have studied quite a lot in the lab is how this mutation rate is variable also at more local scale. I'm not going to enter in detail to that, just to tell that there are very interesting patterns. And with this type of analysis, we can learn a lot about the interaction between DNA repair and proteins binding to the DNA and chromatin, and even how the structure of DNA affects mutation rate. I think I'm going to give only the general idea, that is that there is an interplay between DNA damage, DNA repair, chromatin conformation, structure of the DNA, proteins binding to the DNA, nucleosomes, which is actually very interesting to understand, and is also important to bear in mind that not all sites have the same probability to mutate, and when we try to go to the next step, which is understanding selection, this needs to be taken into account. So when we study selection, the main question we have is which mutations are drivers, at least in our lab. And because tumor genomes contains thousands of mutations, when we sequence the whole genome, identifying the few of them that are drivers is not an easy task, especially we have one tumor, but the way we approach it is to analyze hundreds of tumors in a cohort and identify signals of positive selection in the pattern of mutations. And with this, we and others have seen that this is a very effective way to identify cancer genes. For example, we look at the mutations in a tumor and see if there are more mutations than expected in the gene in a cohort of tumors compared to neutral mutagenesis. That's why we have to take into account how the mutations occur in different regions of the genome and in different tree nucleotides. We can also ask if there are mutations clustered in particular regions more than expected, or if there are more functional impact mutations in the gene than expected. All these are a number of methods, some developed by our lab, some developed by other labs. Like, for example, DNDS was developed here at the Sanger by Íñigo Martín Corena, and we have developed others of these methods. We use 7 methods, the ones that you see listed here in the pipeline of Intogen. And this is a pipeline to take a cohort of tumors and identify cancer genes based on these signals of positive selection. We apply this to 33,000 tumors in 73 different cancer types, and using the mutation pattern analysis to identify positive selection, we identify 619 cancer genes. This is what we call the compendium of mutational cancer genes, and this is in Intogen. Intogen.org, you can explore and download all the data. Most interesting thing is that this is a very effective approach. Only using the pattern of mutations, you identify most of the well-known cancer genes. If you think of any cancer gene, it will have these signals because the signals are very clear. But because it is an unbiased analysis, it can also identify new genes and also identify new interactions between genes that maybe we knew that were important in lung cancer, and now we see that are also important in 3 other tumor types because we find these signals. One thing is identifying the cancer genes, and the next step is to identify which specific mutations in these cancer genes are driver mutations. This is published. I'm not going to explain it today. It's just to say that we develop machine learning approach inspired by evolutionary biology, in which we also play with this variation and selection, observed mutations and expected mutations by neutral mutagenesis. And with that, we develop a model for each of the genes in each cancer type, in the cases that we have enough mutations, that can distinguish between driver and passenger mutations. And then once we have one of these models, for example, TP53 in colorectal cancer, we can apply to all the mutations, all the possible mutations in cancer gene, and generate a map like this that tells us where are all possible driver mutations, including those that we have never observed in tumors. So summary of this part has many things that probably many of you already knew, but it was important for me to explain it because it relates to the second part. And I show you that some chemotherapies leave a mutational footprint in normal tissues, that positive selection signals identify cancer genes, and in silico saturation mutagenesis of cancer genes with BOOST-DM can identify the driver mutations in these genes. Okay, going back to the beginning. I told you that our body is a mosaic of clones, and it has only been recently that we as a community, have been able to see these clones. Maybe one of the very first interesting results was this one by Íñigo Martín Corena here at the Sanger, in which it was made evident that these clones with driver mutations and with positive selection are in the normal skin by studying the eyelid. Another very clear case, very well studied as well, is clonal hematopoiesis. These are stem cells in the bone marrow that acquiring specific mutations may acquire a selective advantage to grow farther and more than neighboring cells, leading to clonal expansions in blood that it's called clonal hematopoiesis. When this clonal hematopoiesis can be detected, it has been seen to be related to an increased risk of leukemia, for example. also cardiovascular problems and other types of diseases. I want to remark that this is a healthy condition. Clonal hematopoiesis is not a disease, but is associated to, for example, an increased risk of leukemia. So we were interested in start studying somatic mutations in normal tissue, and the first opportunity was somatic mutations in blood. I told you that most of these TCGA, Hardwick, and others, because they need two samples from the same patient and blood is the easiest to obtain, most of them we have as a second sample blood. This gave us the opportunity to identify somatic mutations in blood doing a reverse mutation calling. So we try to identify somatic mutations in the blood using the tumor DNA as germline control. And we could see that we can identify these somatic mutations. And once we identify somatic mutations in a normal tissue, in this case blood, if there is positive selection, we can use all the technologies, all the techniques that we have developed before to study cancer. And in this case, we use the pipeline of Intogen to identify not cancer genes in this case, but clonal hematopoiesis driver genes. Right, applying the pipeline into the somatic mutations in blood, we can identify genes that have positive selection in the blood somatic mutations. And this way we identify all the usual suspects of clonal hematopoiesis, but we can also identify some others that were not that obvious, and they also have these signals. Once we have these clonal hematopoiesis genes, we can also apply BoostDM. BoostDM that I mentioned it briefly, that generates this machine learning model to identify which specific mutations are drivers. Now we can apply to clonal hematopoiesis, and for example, in the case of DNN3CA that we have plenty of mutations, we have a lot of power to generate very good models of BoostDM that in this case identify which mutations in DNN3CA are capable of driving clonal hematopoiesis. There is a paper in bioRxiv that you can read already, but the paper is now in press and it should appear very soon in Cancer Discovery. Okay, I told you before about the effect of chemotherapy as a mutagen creating mutations in the DNA. We wanted to study a bit better what is the effect of chemotherapy in our cells. And Uriol analyzed the effect of chemotherapy in clonal hematopoiesis. The clonal hematopoiesis grows with age, so the older we are, more likely we are to detect clonal hematopoiesis. We saw that it also grows with in the cases that patients have been treated with chemotherapy. Right, the probability to identify clones in the blood, to identify clonal hematopoiesis, is higher in treated patients. This has also been seen by independent cohorts by other people, so it's a very robust result. And it's not only that there is increased clonal hematopoiesis, but is a clonal hematopoiesis of a particular type. For example, clonal hematopoiesis of mutations in CHEK2, PPN1D, or TP53 are the ones that are more associated to chemotherapy, while it's different to the genes that we see more often associated to age, like, for example, TET2 or DNN3CA or others. So it's basically that the chemotherapy, so think about it. Chemotherapy is a very strong treatment that kills many cells, especially on tissues that are highly dividing, and hematopoietic tissue is one of them. So many cells in the hematopoietic tissue die during the treatment, and some survive. The ones that have mutations in CHEK2, PPN1D, TP53 that are genes related to coping with DNA damage or DNA damage sensing, they survive better. And that's why they lead to clonal expansions, right? This is the rationale behind that. So when we wrote the paper, this is our explanation. Chemotherapy provides selective advantage. We also saw that these mutations were not new mutations created by the chemotherapy, but there were mutations that were pre-existing in the blood from before. So pre-existing mutations, upon treatment to chemotherapy, what chemotherapy does is to change this environment very strongly, selecting cells that survive better under the chemotherapy. And this leads to this clonal expansion and may increase the risk of leukemia. Okay? Clonal hematopoiesis is linked to the increase of leukemia. At the same time that we were exploring these results, there was this paper published. It's a paper by Alan Balmain and Dave Adams in which they studied the tumors of mice treated with carcinogens. The idea was to see if these tumors had a specific mutational signature of the carcinogen. That was part of the Mutograph project. Surprise was to see that out of the 20 carcinogens that they studied, 17 of them didn't leave any footprint, any mutational signature, and they didn't show any increase of mutations compared to spontaneous tumors in the same tissues. So it seems that many of these carcinogens are not mutagens. We were invited to write news and views, and this, together with our results on chemotherapy, actually made us think a lot. And this is the figure that we wrote in for the paper, that we said, okay, carcinogens, or what we had in mind, many of us, is that many carcinogens are mutagens, and the way they increase cancer risk is by creating new mutations. Some of them may be drivers, and this way lead to increased number of driver mutations that may lead to clonal expansions and eventually to cancer. But now we are seeing that actually many carcinogens are not mutagens. And mutations are still essential, but mutations are already there. And are already there just because many mutations occur every day in our cells without the need of specific mutagens. So all the variability is already there in our tissues. And what the exposure to a carcinogen does is to change the environment and change the selective constraints, leading an advantage to pre-existing cells with pre-existing mutations and increasing and leading to a clonal expansion. So this way, carcinogen can increase the number of cells with driver mutations without creating new mutations. And this way also increase the risk of cancer. So that's what we wrote at that time. And later on, we had the opportunity to discuss with Alan Balmain. And in our discussion, basically, Alan told us, this model has a name, and it was proposed more than, or about 80 years ago. Right? This is the model of two-stage mechanism of carcinogenesis proposed by Berenblum and Shubik in 1947. And this is called cancer initiation and promotion. And this can be equated to what we were saying, basically, and what we are observing in all these results, that mutations are essentially unimportant. And this is in the first step, initiation, a cell acquires a potential driver mutation, but maybe there without leading to any clonal expansion until there is a second step called promotion. And promotion is a change in the tissue environment, modifying the selective constraints and providing this initiated cell a selective advantage. And this is the moment where there is a clonal expansion, and this increasing the risk of cancer. Okay, so initiation and promotion, and if we think on these terms, a carcinogen, that is an agent that increases the cancer risk, can do it in two different ways. One is by increasing the number of mutations, that would be a mutagen, or changing the environment, providing these initiated cells with a selective advantage, and this would be a promoter. A carcinogen can be a mutagen, can be a promoter, or some carcinogens, like for example, the chemotherapies that I mentioned to you, are both. They create mutations and they change the environment and are promoters. The focus of the Cancer Grand Challenge is on promotion. This step is understudied in comparison to the step of mutagenesis that we have all techniques and we can sequence the genome and we can identify mutational signature. There are the Ames test as well. So this is much better studied, and promotion it's much more difficult to study. So we focus the project of the Cancer Grand Challenge with Alan Balmain and Paul Brennan on cancer promotion. And these are the two questions that we are asking. We want to study the clonal composition in normal tissues and ask how the exposure to promoters and risk factors in a person, we are also doing it in mouse and organoids, how the exposure to these promoters shape the clonal composition of the tissues. And the other question is how the clonal composition of the tissues relates to cancer risk. For example, in the case of clonal hematopoiesis, it relates to leukemia. Does the clonal composition in other solid tissues relates to cancer risk? I'm going to show you very preliminary results of Bladder. And here we are asking about the clonal composition of bladder urothelium, and we are asking how this relates to risk factors like age. Males have higher risk of bladder cancer than females, smoking, or previous chemotherapy. This is very preliminary work. I'm very happy to share it with you. Please do not post it because it may change still a bit while we are working on it. But I'm very happy to share it with you and also to have feedback if you have it. Before we started this study, there was a very important study that paved the way, let's say. And this is the study of Lawson et al., led by Íñigo Martín Corena, in which they did laser capture microdissection of bladder urothelium in 15 donors and targeted exome and whole genome sequencing in some of these samples to identify mutations. They could see, these are the two main results from my view. There is a widespread positive selection in the bladder urothelium, and there is inter-individual variability in the mutation burden and in the selection. So different people of these 15 donors have a tendency to select different clones with different driver mutations and also to have different mutation burden. So this is the sampling that we did. It's a different type of sampling. Instead of laser capture microdissection, we did a sampling that is a broader sampling with brushing of the two regions of the bladder, the dome and the trigon. This is work in collaboration with Rosanna Risques in University of Washington. And these are samples that come from autopsies. Once we have this broader sampling, we expect to have clones, but the clones are going to be very small. So we need to sequence with duplex sequencing so that we can identify very rare and low variant allele frequency mutations. We did around 5,000 X duplex depth on these genes that are genes related. These are about 15 genes related to bladder cancer and genes that were shown by this previous study to show positive selection in normal tissue, in normal bladder. And then we analyze all this data. I show you here Raquel and Farriol, who are the two PhD students leading this analysis. And in all this analysis is to identify in a reliable way the mutations that are in this normal urothelium, understand the variation, how the mutations occur, mutational signatures, etc., and measure selection of these mutations. Very overview of the results. We find hundreds and thousands of mutations per sample. Every sample here is a column, and the number of mutations that we identify per sample, you see it in the y-axis. We identify actually thousands of mutations in each gene. With only 77 samples, for example, we have identified 7,000 mutations in ARID1A. And if you compare that to the 15 years of cancer genomics, in Intogen we have 905 tumors. bladder tumors, and we have identified 251 mutations in ARID1A. So we see a much higher number of mutations with this broad sampling. And the reason is simple. Every tumor is one clone. While here, when we analyze normal tissue, we can see many clones. So in every sample, we can see many mutations, and including many driver mutations in these genes. This is just the same plot changing the scale for you to see. And one thing that is interesting is that the frequencies, the relative frequencies of these genes change. And one way of seeing it is like this. This is already corrected by length of the gene and even coverage. So it's mutations per megabase. And you see that, for example, CDKN1A is the most frequently mutated gene in normal bladder. TP53 is among the bottom, the ones in the tail, but is the most frequently mutated in TP53. This is just some food for thought, saying that it's very interesting to see that the frequency of these genes, of the mutation in this gene, is very different in tumors and normals. Although, yeah. So then we analyze the mutational signatures. And in particular, we analyze the mutational profile to use these frequencies as a probability of every 3 nucleotide to mutate. And this serves us to model neutral mutagenesis in a gene. For example, in this gene, we can now go through the gene and generate mutations in silico following neutral mutagenesis. And then we can see that this is the pattern of mutations that we would expect in this gene, in these individuals, in these urotheliums. But the pattern of mutations is actually different. And this is the way, this is just to illustrate what it means to measure positive selection, and is to compare this neutral mutagenesis with the observed mutations and start seeing that the functional impact of the mutations is different, the clustering of the mutations is different, how they occur and where they accumulate, etc. So we have adapted the methods that we applied to tumors now to analyze normal tissue, OncoDrive FML, OncoDrive Clastel, Omega, that is a version developed in our lab for DNDS, and OncoDrive 3D. And we have seen that all the genes except FGFR3 have positive selection, and actually very strong signals of positive selection. This is just the result of Omega. This is the NDS value. Anything above 1, it means positive selection. And you can see that there is a very strong Omega value, especially for truncating mutations in these genes. Now we can use this Omega value, the DNDS value, per gene and per sample. And we start seeing this inter-individual variability. Every column here is a sample or one of the urotheliums, and every row is a gene. And you can see that, for example, these urotheliums here have a preference to select KDM6A, have a very strong DNDS value for that. These ones, much higher preference for TP53. These two ones for FOXQ1, while the others, FOXQ1 is very low number of mutations and no signal of positive selection. So this inter-individual variability is very interesting. And the question is what it relates to. So what these people have done, have been exposed to, that this urothelium selects FOXQ1 compared to this one that selects TP53. This is a difficult question to answer. That's the question that we would like to understand. For the moment, we are analyzing a number of well-known bladder cancer risk factors to understand where the difference are and how they relate to these features. The first thing we can see is that there is correlation with age. So the older the person is, this is age, the higher the percentage of epithelium that is covered by these driver mutations. Okay, this is expected, and this is seen in clonal hematopoiesis, for example. And the only other result that I will show you is that there is, in the incidence of bladder cancer, there is a difference between males and females. Males have 4 times higher incidence of bladder cancer than females. And we were wondering if there is any difference in the clonal selection in the clonal composition of normal bladder urothelium. And we have seen that there is a very strong difference, especially of these 4 genes. When we look at protein-affecting mutations, the mutation rate is much higher in males than in females for these 4 genes. And this is not the case for non-protein affecting mutations, which means that it's not that there is an increased number of mutations overall, there is an increased selection in these genes in males. I show you the result like this, but we have also done multivariate regression analysis corrected for confounding factors, including smoking, age, and others. And this is a very strong signal that shows this difference between males and females. So I finish here. Quick summary, because I explained you many things. I told you about chemotherapies that act both as mutagens and promoters. Told you about positive selection signals to identify cancer genes, but also to identify clonal driver genes, like in blood or in bladder. BoostDM, we can use it to identify driver mutations, but also cancer driver mutations, but also clonal driver mutations. And there is this extensive clonal selection in normal tissues. I talk about blood and bladder. I show you about this difference in clonal selection in normal bladder urothelial in males and females. And in general, in prominent, we are trying to understand this promotion step, that is the step of selecting specific clones in a specific environment, and how this can inform prevention, because this is before the tumor develops. Want to thank everyone who did the work. I think I mentioned most of them on the way. And we want to thank again Rosanna Riskes and then funding agencies as well. Thanks for your attention.
No, thank you for a fantastic talk. We have time for questions, and I already see the hands going up in the audience. So, James, please.
Hi, Núria. First, thanks so much for joining us despite your illness. It's quite heroic. I hope you get better soon. So the question really relates going back to the Riva et al. Study, which you talked about how critical that was for understanding that many carcinogens aren't necessarily mutagens, but perhaps they're selected. They're promoting selection. And I'm wondering if you or anyone has plans to actually use mouse models, because all of us that are doing duplex-seq have really been focused on human tissues, but to use mouse models, to use a duplex-seq type approach to look at how carcinogens like that actually change selective landscapes, and whether that could be used as sort of a secondary screen for potential carcinogens.
Yeah. Thanks for the question, James. And yeah, again, sorry for not being here. I was hearing myself talking, and I realized that I sound like a bit ill with my voice. So I'm sorry I don't put the camera, because I don't have my best face today. So yeah, but the question of mice, we are doing it in prominent in humans, and this is an example is the bladder study, is kind of a type of study that we can do, but then we are also doing it in mouse. Alan Balmain is treating different mouse with different carcinogens or promoters, suspected promoters, and then profiling the normal tissue. And we think actually that this can be a short assay for carcinogenesis effect, right? Because you don't have to wait for the tumors to develop. That these are very long assays, taking many months, and maybe in only a couple of weeks or so, you can already check the normal tissue with duplex sequencing and see the effect of these suspected promoters. That's what we are testing in the mice.
Great, thank you.
Hi, thanks for a very interesting talk. I've got a question to the bladder data that you showed in the end. Could you speculate on the differences between men and women? So one thing that comes to my mind is that women are more prone to urinary tract infections, but it seems to be a bit counterintuitive that they then have less selection? So did you account for this, or could you speculate on alternative explanations?
Yeah, no, that's a very, very good point. We are thinking a lot about that, and we are happy to have suggestions. We are thinking about things that are different between males and females are hormones. You are totally right that the probability or propensity for infections in the urinary tract are also very different. The composition of the urine is also different, and we were reading some papers about that. The reality is we don't know what is causing this, and it could be a combination of these things. There is this difference between male and female, but then there is also a very strong difference between individuals. So I don't think that with our cohort we are going to be able to solve it all, or because it's still a small cohort to account for all these differences, but we hope to at least advance a bit. Yeah.
Hi, Núria. Thank you very much for this nice talk. This is Christoph Plass from Heidelberg. I was also intrigued by the bladder cancer data and the high or increased risk in male patients or in males. And if I remember correctly, there are studies showing that there is loss of the Y chromosome occurring in blood samples that correlates and associates with higher risk. Is there anything that you find that may link to a loss of Y or increased mutations in genes on the Y chromosome?
Okay, so you mentioned loss of Y in the blood or in the bladder?
Yeah, in both. I mean, these are epidemiological studies that have been published about 10 years ago, showing that loss of Y, or mosaic loss of Y, it's called, is a risk factor for bladder cancer, for lung cancer, for many cancers. And then in the tumors itself, you see also an increased loss of the Y chromosome.
Okay, no, that's very, very interesting. No, I have to think if we can check, how we can check this in normal tissue, because duplex, we are not able to see this. And identifying something that is rare in terms of copy number, it's not easy, right? Because identifying copy number alterations in a low proportion, we don't know how to do it. But yeah, no, it's a very interesting thing. These 4 genes are, if I remember correctly, only one of them is in the chromosome X, or 2 of them, but the others are not. And there are other genes in the chromosome X that are not positively selected, so it's not explained by being or not in the chromosome X. Maybe a bit of it's related, but it's certainly not the story. Thanks.
We have one from online here. Aman says, "Thank you for your presentation, Núria. I have a question that's not related to your talk. I'm curious about ribosomal protein truncating mutations and their potential role in promoting cancer cell progression. Can you share your opinion on this? Do you think these mutations are helping in cell selection?"
Ribosomal, okay, so, because there are many ribosomal genes and they are very repetitive, most of us tend to ignore them because they are difficult to identify, even the mutations, which doesn't mean that they are not important and interesting. Actually, they may be super important and super interesting, and I think someone should be checking that because it's overseen at the moment.
Hi, Núria. Great talk, great work. I had the question again on this discrepancy between men and women in bladder cancer. I think you made quite an impression. I was wondering if you had some epidemiological data on eating, drinking, smoking status, as we know there are some cultural differences between men and women. And in case you don't, or even if you do, if you also have data on mutational signatures that would explain the differences, if there is a specific process that could explain the differences in men?
We have data on smoking history, alcohol, chemotherapy treatment. So we have some information, we don't have it all, for example, about dietary things, we don't have any information. Well, we have BMI that can help a bit. And we are including all this in our regression analysis and epidemiological analysis to make sure that when we may do a claim, it's not confounded by other things or where is the signal. As I said, this is still a small cohort. I mean, I would like to have like thousands of these, and then we would be able to tease apart the effect of different factors into this clonal selection. Now, I think we are powered to see the bigger effects, which to me, male and female, looks like a very big one, not confounded by all the others that we have.
Good. Maybe let's make for the microphone to arrive.
Hi, Núria, thank you for your amazing talk. I'm just thinking, from a clinical perspective, at medical school, we're taught that schistosomatitis infection can significantly increase your risk of getting squamous carcinoma of the bladder. I don't know how many, if you have that data of what kind of infections your patients have had, or what kind of ethnic background they came from. But if there was a large African population who had schistosomatitis infection, that might be interesting to explore as well, because from a clinical perspective, we're taught that can massively increase your risk of getting bladder cancer.
Mm-hmm. And one question to you. I think this is similar to the first question, right? Higher chances in women than in men. I think this part of the infections is interesting. Somehow it could go the other way around, and the risk of cancer, because the risk of infection is higher in females, but the risk of bladder cancer is higher in males. So it may have something to do with increased risk in some people. But I still don't know how to explain the difference between males and females. So I think it's a very open question at the moment, and all these inputs are useful.
Okay, other questions? So maybe I also ask one, which is very general. So if I understand the collection of your data on normal tissues correctly, one finds mutations and also microscopic clonal expansions almost universally across tissues. However, cancer incidence varies quite substantially between organs. So what would you suspect is the reason for that?
So between the difference on increase of clones in tissues and developing cancer, or cancer incidence across tissues.
Yes, is there a relation, or if not, what is the reason why this relation may not be one-to-one?
Okay, yeah. Oh, that's... So every tissue is completely different, and so the way I usually see it is more... John Carnes already in 75 wrote a very nice paper where he talks about the structure of every tissue and how this is in part shaped probably as a barrier for cancer development, right? For example, colon with all these crypts and the way that the stem cells are placed, and because this is a very dividing tissue, it's a very risky tissue, and so it's probably about the structure of the organs and the tissue, and this already creates barriers to tumor development, so full uncontrolled growth. And these barriers are different in different tissues because the structure is different, like in skin or esophagus. You have all the stem cells in the basal layer, and then there is no limit, so the clones can grow around in this to this surface. So how I think all this relates a lot on the probability of every tissue to have a full discontrol towards the tumor. But I don't have a specific answer on how I would measure that or how I would do it. Yeah. It's a good question.
But thank you for sharing your thoughts on this. This is certainly thought-provoking. If there are no...
We're at time, so...
Yes, so if there are no further questions, then since we're also right on time, let's thank Núria again for really a stellar presentation. We all hope you get better soon, and to welcome you next year for the next iteration of the meeting, hopefully.